Okay, this is the lecture for chapter 11, which is the nucleus. All right, so just a quick overview, and actually not very long of a lecture, only about 50 slides. So we'll talk about the nuclear envelope, right, which is going to enclose and surround the nucleus, and traffic between the nucleus and the cytoplasm, right, things moving in through and out of, and then organization of chromosomes, and we'll talk a little bit about nuclear bodies and different types of nuclear bodies. Okay, so we all know a little bit about the nucleus, right? It says it's the main feature that distinguishes eukaryotes from prokaryotes. Remember that prokaryotes do not have a nucleus, right? No membrane-bound nucleus. Houses the DNA, and obviously it's the cell's control center, okay? It says that separation of the genome from the site of mRNA translation plays a central role in eukaryotic gene expression, right? Meaning that you have to transcribe those, the DNA into mRNA, then it needs to be transported out of the nucleus before it processed first, right? Then transported out. And so there are many, many steps in there in which regulation can occur, right? And so that's sort of what it means. Anyway, and we've talked about all of those things already. Okay, so let's talk about the nuclear envelope first. So it separates the nuclear contents from the cytoplasm, right? It also controls the traffic of proteins and RNAs through nuclear pores, right? And so there are pores, which are essentially openings through the nuclear envelope. And we're going to talk a lot about them and talk about the movement of things through that nuclear envelope. Okay. So again, it says plays a critical role in regulating gene expression, right? If an mRNA that was made inside the nucleus is not able to get out of the nucleus, it would never be translated, right? And so what's getting in and out is going to affect gene expression. Okay, so in terms of what the nuclear envelope consists of, so two nuclear membrane, right? So it's a double membrane. Also, something called a nuclear lamina that we're going to talk a little bit about that sort of underlies that inner membrane, and also nuclear pore complexes. So the envelope consists of these three things. Okay, the outer membrane of the nuclear envelope is actually continuous with the endoplasmic reticulum, the ER. Okay, there's space obviously between the inner and the outer membranes, and that's actually directly connected with the lumen of the ER. So basically, again, that envelope that surrounds, or particularly the membranes that surround the nucleus, are actually continuous with the endoplasmic reticulum. Remember we said, we talked about the ER before, I guess briefly in the beginning of this course, but also in general bio, that the endoplasmic reticulum and the Golgi, right? Those are all membranous types of organelles, okay? All right, the inner membrane says it has integral proteins. And so remember, if we just think about the plasma membrane, integral proteins are going to be proteins that span the entire bilayer, right? And so some of these integral proteins will actually bind to the nuclear lamina. Okay, so you can kind of think of the nuclear lamina as being analogous to the cytoskeleton, and actually it attaches to the cytoskeleton too. So if you can kind of think of, right, so if you think of the plasma membrane, right, and that double membrane, sorry, not double membrane, bilayer, right, double phospholipid bilayer, not a double bilayer, a single bilayer, right, which is two layers of phospholipids. I don't know why it's so difficult. And you have integral proteins that span through that, right? And then remember, those integral proteins will connect or be bound to proteins that are part of the cytoskeleton. So it's kind of the same idea here. You have proteins that will span through that inner membrane of the nucleus and actually be connected with the nuclear lamina, okay? All right, so this is just showing you a picture here of that nuclear envelope, and so you can see the chromatin in here, you can see the nucleolus. We'll talk about that towards the end of the lecture, and this is just pointing to those pore complexes, right? And so these sort of looks like openings through the membrane where things can be transported across. Okay, and so if you zoom on this a little bit more, this is actually an electron micrograph, right? And you can see here's the inner membrane of the nucleus. This is the outer membrane. You can see that it's continuous with the endoplasmic reticulum, and then here it's showing you the pore complex. Okay, and so if you look at that in a drawing type of diagram here, again, you can see this is inside the nucleus, right? Showing you the nucleolus. Here is the pore complex, outer membrane, inner membrane, and you can see that the outer membrane is continuous with the endoplasmic reticulum and is pointing out some ribosomes that are sitting on the ER, right? Because that's the rough ER. And you can see there's some space in between the inner and the outer membrane, okay? And so we're going to talk, at some point we're going to basically talk about kind of all these things that are part of that diagram. Okay, so nuclear membranes, just like the plasma membrane, like I mentioned, are phospholipid bilayers. They are permeable only to small non-polar molecules, okay? Just like any other phospholipid bilayer. And so the pore complexes are the only channels that small polar molecules, right, ions and also macromolecules can move through, right? So if you're talking about transporting an mRNA out, it's going to have to move through those pore complexes, okay? So everything that's moving in and out will go through those pore complexes, with the exception of small non-polar molecules, which would be able to cross that bilayer on their own. Okay, so now this is showing you this is an electron micrograph again, but this is showing you the surface here, right? And you can actually see those pore complexes. All right, so we haven't talked yet about the nuclear lamina. We mentioned it, right? And I said it's sort of analogous to the cytoskeleton in that it provides structural support. So you can think of it as a fibrous sort of mesh that provides structural support to the nucleus, right? And the reason why it's called lamina is because it consists of proteins, fibrous proteins, meaning long, right? Fibrous proteins called lamins, and also other proteins as well. Okay, so if this is an electron micrograph, and it looks like a wire mesh sort of material, right? And so these are the lamins, they're fibrous, and other proteins as well that make up that lamina. Okay, so let's talk a little bit more about the lamins themselves, okay? And so, again, they're proteins, they're fibrous or they're filament proteins, and they will associate together to form higher order structures, okay? So two lamins will interact with each other to form a dimer, okay? And I'll show you a picture of it. So the alpha helical regions will wind around each other, and they sort of form this coiled coil. Those dimers, right, those two lamins that are together, so two lamins together is a dimer. Dimers will then associate with each other to actually form the lamina. And so you can see that sort of provides strength, right, more strength in that structure. So again, this is one single lamin, right, that dimers form, they sort of wrap around each other. And then these dimers then associate, kind of shows you this sort of head to tail association with each other to make a longer filament, and then another, then they sort of line up side by side, and this is sort of the higher order structure, and that's why you see this mesh-like sort of framework for that nuclear lamina. Okay, so the lamins, as I kind of said before, right, the nuclear lamina will be obviously bound or sort of attached in some way to the inner membrane, right? So lamins will bind to inner membrane proteins, and some of these, and we said integral proteins. And so an example of one of those proteins that spans through that inner membrane of the nuclear envelope, right, is called, these are some examples, emerin and lamin B receptor, okay? And those will then bind to the nuclear lamina, right, to those lamins that are in the nuclear lamina, and that's what's going to provide that sort of structure and that framework, okay? They then, like I said earlier as well, they then are connected to the cytoskeleton. So imagine it's sort of anchoring in there, right? And so we have part of the cytoskeleton attached to the nuclear envelope and to the lamins, and to the nuclear lamina, okay? And so that again provides that sort of structure and sort of anchors everything in place, okay? It says lamins also bind to the chromatin. So you can see that nothing's really just kind of floating around, right? Both inside the cell itself, but also even inside the nucleus, right? That lamina is there to provide that structure, and it's going to basically bind to or attach to the chromatin that are inside the nucleus. Okay, So this is showing you an example of that, right? So here's your nuclear lamina, right? Showing you kind of all in here. And it's just showing you how now this is sort of associated via these proteins that we mentioned, okay? So here's your inner membrane, here's your outer membrane, here's a pore, right? You can see some ribosomes sitting on here because basically we said that outer nuclear membrane is basically continuous with the endoplasmic reticulum, okay? So that's why you see that. You can see here, we mentioned this lamin B receptor, right, LBR, and we mentioned emerin. So these are integral proteins that span through that inner membrane, and they also attach to the lamina, right? So the lamina is in yellow, okay? And then it's also showing you chromatin in here that are also attached or associated with the lamina. And so you can see the same thing here. Then if you look at these proteins, right, this sort of complex, right, this link protein complex that's mentioned there, you can see that, right, this one is going to span through the inner membrane, this through the outer membrane, and then it's going to attach to the cytoskeleton, right? And so again, this provides that, that framework and that strength. And again, anchors sort of everything in place and keeps things where they should be, right? Okay, now, one sort of interesting thing is that mutations in lamin genes actually cause several inherited particularly tissue-specific diseases, which is kind of interesting because if you think of the lamins, or you think of the nuclear lamina, you obviously have the nuclear lamina in every cell type, and you would imagine that it would be just as important, in a muscle cell as it maybe is in a skin cell, or a lung cell, right, or a heart muscle fiber or something like that. But there are actually differences in terms of, like I said, if you have a mutation in one of those lamin genes, you do see these sort of tissue-specific diseases. And so one of these is showing you a picture here. And I actually, one second, I'm going to kind of get off of the, in fact, this is a different picture than is in the article I'm going to show you for 1 second. So on Moodle, I did post this. So this is actually from your book. So I just want to kind of draw your guys' attention to this. And yeah, you probably still can see this. Okay. So that's the same diagram we went over, right? And so there's a little summary about some nuclear lamina diseases. And I said they're tissue specific. So I definitely want you guys to take a look at this. Okay. So muscular dystrophy, right, is essentially linked to a lamin gene, okay? So I want you, again, I'm not going to sit here and read it over or read it for you, but I want you guys to read this, and maybe we'll, hopefully I'll remember to kind of bring it up when we meet next week. But definitely something you should be familiar with, and I could definitely kind of ask you an essay question that's sort of related to this, okay? So this picture here is a child with Hutchinson-Gilford progeria. So I think I asked you kind of about progeria before. I don't know why, but anyway, there have been I've seen things on TV like specials about this disease. It's a pretty rare disease, right? And so what happens in that disease is you basically have expedited, the aging process. And so this is showing you a child that looks like an older man because of this progeria. And so this has been linked particular to a particular lamin, right? So let's see what it says here. Yeah, so this is the deletion, right? So the mRNA encoded by the mutant gene shown has 150 base pair deletion, which is shown in dark blue here, in exon 11, right? So it's showing you the exons of that gene, right? So this is the gene for lamin A, and in fact, they've actually localized that mutation and know exactly what exon it's in. And that's leading to this disease. And so, again, this article talks a little bit about some other diseases that are linked to the nuclear lamina as well. So again, definitely take a look at it. It's posted on Moodle. All right, so I'm going to go back to the lecture. Okay, so this picture was just from an older version of that article, but the updated one is the one I just showed you. Right, same thing. It's basically same information that's in there. This is what I just drew your attention to in the article. Right? So like if I asked you about this, of course I'm not going to expect you to, I don't expect you to memorize where the deletion is or anything like that, but I may sort of ask you a sort of application based question about this. Okay? All right. Oh, and again, forgot to remind you, make sure you're writing in your review sheet here. So let me just take a look kind of where we're at on that review sheet, right? So obviously we're in chapter 11 now. Yeah, so we talked about, right, nuclear lamina, lamin structure, the associated proteins with the nuclear lamina and how it links to the cytoskeleton, right, and those proteins that are within that inner membrane as well, and then some diseases associated with the nuclear lamina. And so that's really based on that article that I just showed you, okay? All right, now the next topic is to talk about those complexes, okay? So the nuclear pore complexes, they're large. So about 30 proteins actually make up that nuclear pore complex. Again, just to remind you, if we talk about, transcription or RNAs that are synthesized in the nucleus, they have to be able to be exported out of that nucleus to the cytoplasm to find a ribosome, right, so that they can be translated. Also, there are proteins that are needed inside the nucleus, right? And so they have to be imported from synthesis sites in the cytoplasm. Think about all those proteins involved in transcription, right? They have to be able to make their way in. Okay, so it says molecules can pass through those pore complexes by two mechanisms, right? Passive diffusion, easy, right? Small molecules will just pass freely in either direction, moving along their concentration gradient. However, there also is a lot of selective transport, okay? So proteins and RNAs, like we talked about on that last slide, have to be selectively transported, and that actually requires energy, okay? And so this is just showing you this, right? So you got some passive diffusion of small molecules that goes on, but proteins coming in and also proteins going out is selective transport. RNAs coming out is selective transport, okay, and requires some energy. And actually, it's a pretty involved process. We're going to talk about it. Okay, if you actually look at the structure of the pore complexes, and you can look at it using electron microscopy, there are 8 subunits, okay, that are all organized around a large central canal. And then you see there's 8 spokes that are connected to rings at both the nuclear and the cytoplasmic surfaces. Okay, I'll show you a picture, and you kind of already saw, probably maybe remember kind of that structure when we just saw the picture before of the nuclear envelope in the lamina. That spoke ring assembly surrounds the central canal, okay, and then there are other protein filaments that will extend from the rings that kind of form this basket-like structure, right? And so this is actually showing you that electron micrograph where you can see those subunits, right? So this is kind of showing you a sort of top-down view, right? So like this is the central canal going into the nucleus. So if we look at a cartoon of that, this is cut in half, right? And so it's showing you 4 of these subunits, but then obviously it would go all the way around here. And so this is sort of this basket-like structure that they were talking about before. And you can see you have this sort of ring assembly here and another ring or sort of ring assembly, I guess, here, right, on the nuclear face of that inner membrane, and another ring assembly that's facing out, okay? Bottom line is this is a pretty complex structure made up of a lot of different proteins and sort of very specific in terms of its structure. Okay, again, I don't expect you guys to memorize every component of the nuclear pore complex. That's not the point, right? It's just to illustrate that there are many different proteins that make up this pore and to show you the basic structure of it. Okay, now we have to talk about details in terms of how proteins enter the nucleus. Okay, we said it's selective, right? So how is that selective and how does that work? And so proteins that must enter the nucleus or need to enter the nucleus because that's where they function, right? They actually have nuclear localization signals, okay? So there's particular amino acid sequences on them that serve as nuclear localization signals, okay? Those signals then get recognized by a particular special receptor called the nuclear transport receptor, okay? So if a protein does not have this nuclear localization signal, it will not be imported into the nucleus, okay? Likewise, if there is a mutation in that nuclear localization signal, that protein will not make its way into the nucleus and thus will not be able to function because its function is inside the nucleus. It is important that it gets to the compartment that it needs to be in, right? Or where it functions, right? Okay. Nuclear localization signals are recognized by receptors, which are called importance. Okay, so that's what it said here. Okay, so the particular name of that receptor is called an important, which makes sense because it's going to import that protein into the nucleus. Okay, these importants actually work in conjunction with GTP binding protein RAN. Okay, remember we talked about you know, the importance of proteins binding to GTP and GDP, and that regulates their activity. Okay, so here's another example of that. All right, so that RAN protein is super important in controlling the way that that protein is going to move, the direction that it goes. Okay, so there's a lot of words here. I realize there's a lot of words in all these slides in this lecture, more than usual, I feel like. These words basically describe what's happening on this slide here, okay? So importance will bind to that NLS, that nuclear localization signal, and then to the nuclear pore proteins, and basically that complex gets transported across the membrane, okay? And so let's just look at the picture. Again, if you want to kind of check back with the words here, what's going on, you can, but I think it's probably better to just look at the picture and sort of follow along what's going on here. So here's your cargo, here's your protein, okay? This is the protein that functions in the nucleus, therefore it needs to get into the nucleus, and it has a nuclear localization signal. That signal is recognized by this receptor, right, this important. Okay, and this whole complex then will go to that pore the nuclear pore, right? And will enter into the nucleus. Once it gets inside, you want this important to release this protein so that it can go and do its job, right? And so that release is controlled by this RAN protein. Okay, see that here? So showing you here, this is RAN GTP, and then basically that important binds to that RAN GTP, and then it can go out, okay? And so you can stop and think for a second why it's important, not to be redundant, right? Why is it important that the important gets out is because it functions out here. It needs to get another cargo protein and bring it in. If it's stuck inside the nucleus, well, you're not going to have any importance outside to bring it in, right? And so it's super important that this continues so that this can get back out to take another protein in, okay? So this RAN protein is important in moving this important, okay? Another thing that's happening, there's another protein here, okay? This RAN gap, and what that does is sort of control what's going on over here, right? So again, it's also important that that RAN can get converted back to its GTP form, right? So here, once it came out and basically passes this RAN gap, it gets converted into this RAN GTP, which allows that important to be released, right? So this guy needs to be able to get back inside, okay? And that is via this NTF2. So this protein brings him in, and then once he gets it back inside, it can go back, it get converted back to GTP, right, by another specific protein here, and then the cycle can continue. So you can kind of see how all three of the, well, I guess you can kind of break it down to kind of three main components of things that are happening here, right? The important binding the cargo protein and bringing it in. Then this RAN GTP binding to the important and bringing that out. And then that RAN GTP got converted to RAN GDP, and now it binds to the NTF2 and gets back in. Okay, and so all of these, or every single step here, or every single sort of process that we just talked about is necessary, right? If one is not working correctly, or if there's a mutation in one of these proteins, this whole process is going to stop, right? That protein would never be able to be transported into the nucleus. Okay, so super important that you guys understand this process. Again, we're not memorizing this, okay? We're understanding this. And so I put a lot of times on the exam, I'll tell you straight up, is I put the picture up there and I ask you to explain something, or I say what would happen if there was a mutation in this protein, right? What would happen if there was a mutation here, and ask you how the whole process would be affected, okay? So make sure you do understand that. Okay, so that was protein import, right? So obviously things need to get out of the nucleus as well. And so if we want to talk about protein export from the nucleus, they get targeted for export by amino acid sequences that are called nuclear export signals, okay? And they are recognized by receptors or recognized by proteins called exportins, okay, which will bring that protein out of the nucleus to the cytoplasm, okay? It says exportins form stable complexes with cargo proteins in association with RAN-GTP. So again, similar proteins involved, okay? In the cytoplasm, GTP hydrolysis and release of RAN-GTP leads to dissociation of the cargo protein. So again, very similar mechanism, okay, as to what we just saw, right? And so that's what this is showing you here. You got your nuclear export signal, okay, on your cargo. So here's your cargo protein, right? Nuclear export signal. This is the exportin. It binds to it in association with this RAN-GTP, goes out. Cargo proteins released when this RAN-GTP becomes hydrolyzed to the RAN-GTP. And then again, you got another protein here, this NTF2, talked about before, which is actually going to help bring that exportin back in. And in the process, we're going to have, conversion back to the GTP form. Okay, so super similar in terms of mechanisms that were occurring. Again, I don't expect you to memorize this, but if I showed you this, you should know what's going on, and you should be able to tell that this is a protein being exported out, okay? And know that an exportin is that receptor that's responsible for recognizing the nuclear export signal and transporting a protein out of the nucleus, okay? All right, in terms of RNA, so those were both protein import into the nucleus and protein export out of the nucleus. It says RNAs are transported to the cytoplasm as ribonucleoprotein complexes, meaning it's RNAs complex with a protein, right? RNPs. And so the transport mechanism for RNA, take-home message, is not the same as what we just talked about, okay? This is actually showing you, it's an electron micrograph that's actually showing that ribonucleoprotein complex being transported out or sort of emerging out of the nucleus. Showing you that again. Okay, and then now it's fully released. Okay, so like I said, that mRNA transport does not involve those mechanisms that we just talked about, right? It doesn't involve importins or exportins, and it does not involve that RAN protein, okay? There is a distinct, different transporter complex that will move that mRNA through the nuclear pore. And there's a helicase will actually sit on the cytoplasm side and will release that mRNA and again make sure that it moves in that direction. Obviously, we don't want the mRNA trapped in the nucleus. It needs to be able to get out. Okay, so this is showing you the export of mRNA. And so you can see here, right, these are the proteins that are bound to it. So this is that mRNA or that RNP, right, ribonucleoprotein complex. And so it gets transported out. Helicase sort of sits here and basically ensures that moves this way, and basically it'll sort of lead to or sort of catalyze the removal of some of these proteins in that complex to ensure that it's moving that way. Okay, so again, nobody's memorizing anything. We should, if you saw this, I mean, it gives it away. You can see that it's mRNA, but we should be able to explain this or know some basics about it. And again, if I asked you about, what would happen if helicase wasn't functioning in this scenario here, right? Well, maybe, the movement of this mRNA out of the nucleus would not occur. Maybe sometimes it would, but sometimes it may not go out. It may come back in or may get stuck in there or something like that, right? So it is important in this process, okay? All right. And again, we're leaving out a lot of details here, so we just sort of leave it at that. Okay, it says here, regulation of protein transport is a mechanism for controlling protein activity in the nucleus, okay? So it says regulation of import and export of transcription factors is a way of controlling gene expression. Think about it. If there's a transcription factor, well, one, think about what a transcription factor is in your mind. It's what? I mean, it's a protein that binds to the DNA, right? And therefore can inhibit transcription, right? Or can activate transcription. And so if that transcription factor is in the cytoplasm and doesn't get into the nucleus, well, it's not going to have an effect, right? And so the regulation of import and export of these transcription factors actually itself controls gene expression. So it says here, in one mechanism, right, transcription factors or other proteins associate with the cytoplasmic proteins that mask their nuclear localization signal. Okay, so there are other proteins that may block that nuclear localization signal, thus keeping them in the cytoplasm. Again, if that transcription factor is stuck in the cytoplasm, it's not going to have any effect on transcription. It has to get into the nucleus to do that. Okay, so let's kind of continue a little bit more about that sort of mechanism that we just mentioned, right? So it says transcription factor NF kappa B, right? So NF, you can say NFKB, whatever, is complexed with I kappa B in the cytoplasm. Okay, so it says if I kappa B is phosphorylated and degraded, right? Remember we mentioned ubiquitin mediated proteolysis, right? Remember a whole bunch of these ubiquitins bind to a protein, and that indicates to the cell that it should degrade it, right? And it will break that protein down. That was in the last chapter. And so if that I kappa B or IKB, whatever you want to say, is degraded, then that NF kappa B can actually enter the nucleus and activate transcription, okay, of its particular targets, right? If that I kappa B is not degraded, it will be blocking the nuclear localization signal on this protein, and therefore it will not be able to get inside the nucleus to affect transcription, okay? It says other transcription factors are actually regulated directly by phosphorylation, okay? If you guys look at your review sheet, I have a question under it. So we're talking about regulation of nuclear import, right? And it says, what effect does this have on gene expression? And so I'm talking about that right now. So make sure you can answer that question. The other thing I also want to check is what, okay, I'm just checking the chapter 11 homework too. And I think we've pretty much hit on all of them. So just to remind you, it says list the components of the nuclear envelope, describe the components and the function of the nuclear lamina, right? That's pretty straightforward. We talked about that right in the beginning of the lecture. So remember the components in the nuclear envelope, it just says to list them. The inner membrane, the outer nuclear membrane, right? The nuclear pores and the nuclear lamina. Describe the components and the function of the nuclear lamina, right? So we said it's made up of lamin proteins. And what's the function? It's structural, right? Provides that framework. You could talk a little bit about the mesh-like framework or the mesh-like structure. You could talk about some of the proteins that link the nuclear lamina to the cytoskeleton and to the nuclear membrane. For the second question for chapter 11, I'm just, like I said, I'm making sure I hit on these questions. Ask about the nuclear localization signal. And we're talking about that even more now. So if you mutate that nuclear localization signal or you block it for any given protein, it will not be able to enter the nucleus. Okay? All right, so again, be paying attention to the homework questions and to the review sheet as you're going through this lecture. You can just be answering them as you go through. And again, it's sort of helping you remember what's in, I mean, it's all important, right? But helping you sort of remember what I'm emphasizing, right? And making sure you understand those concepts that I want you to understand. Okay, it says other transcription factors are regulated directly by phosphorylation. We talked about sort of regulation of proteins and that you can phosphorylate them, right? A couple times already. Kinases phosphorylate proteins, right? Remember, phosphatases dephosphorylate, and so that affects the activity of proteins. So this is an example saying yeast transcription factor FO4 is phosphorylated right next to its nuclear localization signal, and that interferes with its import because it alters that nuclear localization signal, okay? So again, all ways that you can regulate gene expression really by affecting the import of certain proteins into the nucleus, right? And so this is showing you that here. Okay, again, we need to understand this and understand why this is important on gene expression or how it affects gene expression. Again, that question is in your review sheet. Okay. Okay, so that was the example that I just went through. Here's your NF kappa B, here's your I kappa B. This NF kappa B has this nuclear localization signal on it. However, if this I kappa B is there, it can't enter into the nucleus. Only if this guy gets phosphorylated, ubiquitinated, and gets degraded, will then an important be able to bind to this NF kappa B and bring it in. Okay, here, it's phosphorylation right next to the nuclear localization signal that basically keeps it, or it doesn't allow the important to bind to it because it changes the structure slightly. But if it gets dephosphorylated, meaning that phosphate group is removed, then the important can bind and bring it in. Okay, so you can see how phosphorylation is affecting it here, particularly. The idea is it's not necessarily, so some phosphorylation is involved, but the main thing is that this is going to be degraded, and that, remember, is sort of mediated through ubiquitin, right? If the cell can add a bunch of ubiquitins to a protein, and that targets it for degradation, okay? Kind of two different mechanisms in terms of how this is happening, kind of having the same effect, right? But two different mechanisms. Okay, so the last part of this lecture talks about nuclear bodies, all right? And so even your last homework question asks you to list 3 types of nuclear bodies and describe their function, okay? Particularly the main one we're going to focus on is the nucleolus. And so again, if you look at your review sheet, it says function of the nucleolus and ribosome assembly. That's the one that we're really going to go through in detail. The other ones we're not. We're sort of just going to know that there are many different types of nuclear bodies and just what the just sort of general basic function is of 3 of them, okay? So they're organelles that are within the nucleus, and they basically sort of concentrate specific RNAs and proteins that are involved in a specific process, okay? They're not enclosed by membranes, but again, there are a whole bunch of proteins and RNAs kind of together that are going to function together, okay? All right. So this table 10.2, and I sort of drew your attention to that in the review sheet, is basically just some examples of nuclear bodies. And like I said, we're not really going to go into detail to talk about all of them. The nucleolus is the one that we mention a lot, and you can kind of see it as a darker spot inside the nucleus, right? And sometimes there's not only one, there can be more. And what happens at the nucleolus is ribosomal RNA transcription, right? Processing and ribosome assembly. It's all happening in that nucleolus. And so you can see why that's super important. But again, these are other examples of nuclear bodies, right? So Cajal body, small nuclear RNP assembly, speckle, storage of pre-mRNA splicing factors. What in the heck? Sorry, my children are outside and it's raining. Speckle. Oh, I already said that. PML body. This is another example of a nuclear body. Transcriptional regulation, DNA repair. Okay, polycomb body, gene silencing. So again, in the homework, I asked you to just list 3 and give me just a basic function. So that's really kind of what's in this table. And like I said, we're going to go into more detail with the nucleolus. Okay, but not with any of the other ones. All right. So I said this we just saw this in the last table, but it's functions in ribosomal RNA synthesis and ribosome production, okay? So we all know that ribosomes are super important to the cell because that's where protein synthesis occurs. It says actively growing mammalian cells have 5 to 10 million ribosomes, right? That have to be synthesized each time the cell divides. So you can see why the nucleolus is super, super important. Okay, and so maybe we remember these different types of rRNAs, and remember the S is sedimentation rate, right? It has to do with where it's going to sediment in an ultracentrifuge, okay? It's not any other sort of measurement that we're sort of familiar with, okay? So 5.8S, 18S, and 28S ribosomal RNAs are transcribed as a single unit, right, in the nucleolus by, remember, that one specific RNA polymerase 1. And basically you get this precursor RNA, right? 45S precursor RNA. And so all 3 of these guys come from this same precursor, okay? So this then gets cleaved and you have these. However, this 5S ribosomal RNA takes place actually outside the nucleolus. Okay, so these guys are all transcribed within the nucleolus. This one is actually not in the nucleolus, and it's actually catalyzed by a different RNA polymerase. And we talked about this before, okay, when we were talking about our transcription factor, our chapter on transcription. All right. It says following each cell division, nucleoli, right, become associated with nucleolar organizing regions that actually contain these ribosomal RNA genes. So this is kind of saying, how does this kind of happen in terms of forming that nucleolus? Okay, so it says transcription of that 45S, right, pre-RNA that we mentioned here, pre-ribosomal RNA, leads to the fusion of these small pre-nucleolar bodies, and then thus you end up with this sort of nucleolus. Okay. All right. Okay. So again, they can kind of form these separate little mini nucleoli that then fuse to form a single nucleolus. All right. Okay. Now a little bit more, right? Because we said in the nucleolus, it's transcription of the ribosomal rRNA, but it's also ribosome assembly that's happening there. So there's more to it, right? So the pre-rRNA, right, and also the ribosomal proteins and also that 5S rRNA all need to be assembled, okay? And so ribosomal proteins get produced in the cytoplasm and they get imported into the nucleolus, and then they'll assemble with that pre-ribosomal RNA. Okay, we already said this. I think we already said kind of all of this. I'm going to show you a picture. It'll make more sense. Okay. It says pre-ribosomal particles are then exported to the cytoplasm, and then that's going to yield those two subunits, right? Remember, the small and the large subunit. So like I said, the picture, I think, makes it a little bit easier to sort of conceptualize everything that's going on here. So again, this is a nucleolus, right? This is saying ribosomal DNA, meaning the genes for the ribosomal rRNA, right? The rRNA is here, okay? So transcription is going to happen. You get this pre-rRNA, right? That basically leads to, you know, all of these, well, not all of those, right? The 5S comes in. So remember, you get a pre-RNA. That's referring to this, okay? That 45S pre-rRNA, which is here. Okay, but from there, basically, you're going to then, you know, you're going to combine with this 5S rRNA, which is, remember, synthesized somewhere else outside of the nucleolus because it's actually transcribed by a separate RNA polymerase, okay? But it has to come together here to form that pre-ribosome, essentially, okay? So some of the ribosome assembly is happening inside the nucleolus, okay? Also, ribosomal proteins, okay, are coming in from the cytoplasm. Why? Because those ribosomal proteins were made on ribosomes out here, and then they get imported in, associated with this pre-RNA, and now they can sort of start to assemble to form the two subunits. Okay, so you can see here pre-18S, that essentially then forms that smaller subunit, right? And then here you have the 28S, the 5.8S rRNA, and the 5S rRNA that came from here, associating with some other proteins, right, to form this larger subunit. Okay, and again here, I don't expect you guys to memorize these details. Okay, it is, I guess, a little confusing because there's kind of a lot going on, but you need to be able to explain this diagram like I just did, and so understand what's happening here. Okay, so don't memorize, you know, all the I mean, I guess you got to know some of this stuff, right? But I really don't want you guys to memorize. I guess you don't even really have to worry about knowing 28S, 5.8S. You do need to know, though, that 5S ribosomal RNA does come from another site. It's not actually made in the nucleolus, but that's clear from this diagram, okay? So again, for this, it's mostly just being able to explain sort of how the assembly process of ribosomes happens, right? And what the importance of the nucleolus is, or what the nucleolus is, right? The nucleolus is basically, it's a nuclear body, which you can think of it as kind of an organelle within in the nucleus. It's not membrane bound. However, everything that's there is basically there to function in producing the ribosomes, right? So first, particularly producing the ribosomal RNA. And that ribosomal RNA is made as a precursor first, right? At least some of it, okay? Some of the ribosomal RNA is actually made outside of the nucleolus, right? That 5S, okay? Once the RNA is made, ribosomal proteins come in, okay? All into the nucleolus now, because the ribosomal assembly is happening in the nucleolus. So you have the RNA, the ribosomal RNA associated with the ribosomal proteins, and you basically get the formation of this sort of small subunit, a pre-large subunit here that then gets exported out, and then basically it's sort of final. It's not a fully functioning sort of, or those subunits aren't fully functioning until they're exported out, okay? And so now here you have your 40S subunit and your 60S, okay? But basically all the assembly happened inside the nucleus, particularly inside the nucleolus. Okay? All right. And that's the yeah, I think I hit all the homework questions for chapter 11, okay? And I went through the review sheet as we went through as well. Okay? So make sure you guys understand what's going on here. If you do not, you need to make sure you ask questions next week when we have our Zoom meeting. All right. Bye guys.